my name's Graham Taylor. I'm from the University of Guelph. And if you're seeing this video, it means something has gone wrong with my audiovisual. So I hope you're not seeing this and I'm presenting to you live, but here is a set of recordings as a backup. I'm going to be talking about machine learning for biodiversity. So let me begin by defining what biodiversity is and why it's important. It's the variety and variability of life on Earth. But to scientists, it's a measure of variation at the genetic, species, and ecosystem level. To humans, it means much more than that. As you can see from the list of impacts to the right, it affects our life and the life of the planet in many ways. However, despite its importance, Earth's biodiversity has decreased significantly over the last few decades. In fact, between 1970 and 2016, the population of vertebrate species fell by 68% on average worldwide. And what's causing this global decline? Well, this graphic from the World Wildlife Federation, Living Planet Report 2020, illustrates the biggest threats to Earth's biodiversity and the impact each threat has had globally. The biggest drivers of biodiversity loss are climate change, pollution, invasive species and disease, species overexploitation, and changes in land and sea use, including housing development and unsustainable agriculture. There are places on Earth that are both biologically rich and deeply threatened. These areas have large numbers of endemic species, those found nowhere else, and many of these are heavily threatened by habitat loss and other human activities. Conservation International has identified 36 regions where success in conserving species can have an enormous impact in supporting global biodiversity. These intact habitats represent just 2.5% of the Earth's land surface, but they support more than half of the world's plant species as endemics. Those are species found no place else, and nearly 43% of bird, mammal, reptile, and amphibian species as endemics. In 2017, scientists reported a decline of more than 75% in insect biomass across 63 nature areas in Germany between 1989 and 2016. Now, this is an off-cited study, and it's interesting for a variety of reasons that I'll reference throughout this talk. So for one thing, they study insects, we do too in my lab, and they measure insect decline or biodiversity decline via insect biomass. And so that's something else we do. They employ citizen scientists for data collection, and they use these malaise traps, which look very much like a tent. We're also using malaise traps to acquire data. So for machine learning and computer vision folks, often the first question is, well, what's the data? I'm part of a collaboration led by the University of Jyväskylä, University of Helsinki, and Duke University, and it's called LifePlan. Now, LifePlan aims to build a planetary inventory of life and has enrolled 100 teams worldwide to collect different types of biodiversity data. Now, this is a whole poster for the project. I'm not going to spend too much time on it, but I do want to point out the types of data that's being collected in the project. So there are camera traps. These are capturing images of animals day and night. There's these audio moths that record bird and bat frequencies. There's the malaise traps I mentioned before. These passively trap flying insects, yielding DNA and imagery. There are cyclone samplers that collect fungal spores, which are sequenced to yield DNA. And finally, soil samples are sequenced for DNA of fungi that are living in the soil. Another interesting aspect of the data in this project is it's collected at multiple scales. So there's, as I said, more than 100 sites worldwide that are collecting all of these types of data, but there's also increased sampling in biodiversity-rich areas such as where the project is led in Northern Europe, as well as Madagascar. I'm also a co-PI of Bioscan, led by the University of Guelph, where I'm based, and this is a global interdisciplinary effort to build a biodiversity observation system. It's a $180 million initiative running between 2019 and 2018, and it involves more than 1,000 researchers in more than 30 countries. It focuses on DNA sequencing and a process called barcoding, the use of a single gene on which to base clusters. And these clusters closely match what entomologists call species based on physical inspection. So I'll just give you an overview of the DNA barcoding process. First, we acquire a physical specimen, for example, in one of these malaise traps. Then we perform DNA sequencing on it. This is currently done in the lab, but it's actually moving out to the field using technologies like nanopore sequencers. There's a bioinformatics pipeline to clean up the sequences. And then after these DNA barcodes are assembled, then we can assign a barcode index number through a clustering process. And this gets added to the Barcode of Life database, or BOLD, and along with its taxonomic annotation. So an expert actually physically inspects the specimen and labels it according to Linnean taxonomy. So there's actually close to 12 million sequences in BOLD right now, and 5 million of these have species level identification by an expert. This number grew by 3 million in the last year. And incredibly, a lot of this is manually curated, and at the rate of collection, it's totally unsustainable for humans to be doing this. So it motivates many uses of AI and ML. So apart from the focus on DNA in this project, researchers here at CVPR might be interested that the Center for Biodiversity Genomics is collecting imagery at an impressive rate. So individuals, insects, can be imaged with the K-ONS microscope, as we see here. This collects very high quality images. But we also do a more quicker and common collection procedure, which is known as bulk images. And here, between 100 and 1,000 bugs are typically imaged at once. In some samples, actually, the specimens have actually been imaged both by the K-ONS microscope and in this bulk form, leading to interesting learning problems where we have different kinds of imagery for insect individuals. So one message I do want to convey for the folks at this workshop is that biodiversity yields a variety of interesting machine learning computer vision problems. It's not just apply a CNN or a vision transformer directly to this data. I've actually learned about many different types of machine learning tasks through my work in biodiversity analysis. And this includes things like open world detection, the idea that you might want to discover new clusters or classes after you've trained a model. Also the notion of zero-shot learning from images with using DNA as side information. We might be familiar here with the use of natural language as side information, but these DNA barcodes actually provide a very rich source of side information. This allows you to recognize an individual from an image, even if you don't have that particular species or taxonomic level already in the database. I've also been exploring set valued classification, which I'll talk about in more detail, as well as hierarchical or taxonomic classification. And of course, everybody's talking about self-supervised learning and transfer learning at scale, and this offers the opportunity to build foundation models for biodiversity. So talking about the data in a little bit more detail, biodiversity data tends to have a very long-tailed distribution of its labels. So I'm showing you a plot here analyzing the iNaturalist 2018 data set. And this shows that for if you take the species ranked by their presence in the training data set, you'll see that some of them are very highly represented, more than 1,000 samples for those particular species, but others have less than 10 examples in the data set. When you measure at validation time, you'll see that it's actually balanced. So we're expected to perform well across different species. And so this long-tailed label distribution contributes to degraded accuracy due to aleatoric and epistemic uncertainty, and exacerbates a problem called miscalibration in deep neural nets. So let me dig a little bit into both of these terms. So epistemic uncertainty is the things you don't know. This is the type of uncertainty that we often say is reducible, because by obtaining more training data, we often can reduce this kind of uncertainty in our model. It's often just called the uncertainty. But there's another kind of uncertainty called aleatoric uncertainty that really has to do with things that are stochastic in the world. So this is the irreducible uncertainty. And while typical decision problems might have elements of both types of uncertainty present, there's a fair amount of aleatoric uncertainty in the data, and I'll point this out with some specific examples in our data sets. Now, the other problem I talked about, which is calibration of neural networks, we often interpret the output of neural network classifiers as probabilities. For example, we often use softmax outputs at the very top of our networks. And the notion of calibrated models is really captured by this equation up here. So we like to say when we output that we believe a prediction is assigned to a particular class with a particular probability, we would expect that on average we're accurate to that with that same probability. So for example, given 100 predictions, each with confidence or maximum valued softmax is often used of 0.8, we would expect that 80 of those 100 should be correctly classified. If that is true, then we would say we have a calibrated classifier. But often in deep neural networks, because these are trained with loss functions that are just agnostic to calibration, they tend to not actually represent the right uncertainty in the model. And so this was pointed out by a very influential paper back in 2017 that showed that modern neural networks at the time, ResNets, unlike those like LeNet from the 90s, were actually poorly calibrated. And they showed that even though the accuracy went up considerably from 1998 to 2016, there was a massive leap in average confidence. So the models were in fact overconfident. And when they measured this using something called a reliability diagram that shows the expected calibration error in bins. So each of these bins represents the predictions coming from the model that are between a certain confidence range. So these are the predictions that are highly confident, between 0.9 and 0.1, and these are the predictions that the model is making where it's not very confident, say between 0 and 0.1. And then they look for those predictions at every particular confidence level, how accurate they were. And if the model is perfectly calibrated, of course, we would expect it to line up right along this diagonal here. And they find that for the older models, that's generally true. It follows the diagonal quite well. While we're able to reduce the overall classification error with the modern models, we find that they're not very calibrated at all. And so the point here made by the authors is that when you use these systems as part of real-world decision-making systems, they not only have to be accurate, but these models should indicate when they're likely to be incorrect. And poorly calibrated models don't do that. So a natural question is how about the modern architectures like vision transformers? So there's a paper by Google Brain in 2021 by Minderer and colleagues that revisited this calibration issue in modern architectures, including the vision transformers and hybrid methods like MLP mixer. And they actually showed that they have better calibration off the shelf than convolutional neural nets. And so this is actually positive news that this calibration issue may be something specifically to CNNs. But they demonstrated these results on standard class-balanced data sets. And we've been interested in exploring this in the settings that we explore in biodiversity to see if this holds for long-tailed recognition and under distribution shift too. So I mentioned before, we've been exploring what are called set-valued classifiers. And what set-valued classifiers do is they permit a model to output not a single prediction for classification, but a set of candidate classes instead of one. So from the point of view of the data generating distribution, it's the exact same. We assume that this is not multi-label classification. This is a classification setup where each example X actually has a true classification label Y, but the way that our model operates is different. It's trying to assign an input to a variable size set of labels instead of mapping it to a single label. And the objective here is to do this with low risk, meaning we want to minimize the probability that the ground truth label is not in the set output by our model. And so as I mentioned, it doesn't change the underlying data generating distribution. It's still a classification setup. And it's popular, therefore, to just train classifiers using standard loss functions and then design plugin methods that will adapt a pre-trained multi-class classifier to perform set value classification. So a straightforward approach to doing this is just to provide top K candidates. This is widely used in benchmarks like ImageNet. We often calculate top K accuracy, but there's really no reason to predict a fixed number of classes for every single input. Why do we always have to do top 5? Obviously, the inputs coming in are quite different. Some might be more difficult than others. And so in situations like the fine-grained classification that we do in biodiversity, it's possible to achieve a more informative prediction by relaxing this hard constraint to keep the set size the same for every single input. And so most known set value methods attempt to trade off two quantities. So there's the error rate that the true class doesn't appear in the output set, but there's also the set size. And so I didn't really mention this. You can basically get zero risk by just outputting the largest set size possible. The set size is the exact same as the number of classes, but this is not a really useful set value classifier. So you have to sort of trade these two things off against each other. And so we can make these plots. This is from a benchmarking exercise. There's an excellent set valued classification overview paper survey by Schützen et al. And they evaluate different techniques. And we've implemented the methods that were explored in this survey paper and released some code if you're interested in trying these methods out yourself. But looking at various data sets like IMNIS and ImageNet and PlantClaf 2015, they explore this trade-off. So on the right, on the x-axis, we've got average set size, and on the y-axis, we've got average error rate. So the best classifier would have its predictions sort of towards the bottom left, meaning that it achieves low error while still outputting low, very small set sizes when it makes predictions. And this is a method, a way that we can compare various techniques. And so the red and the orange methods enforce constraints either on the error while allowing the set size to adjust, or on the set size while allowing the error rate to adjust. And then there's also the average control method. And a general trend is that across all of these data sets, allowing individual points to violate constraints, either on the error or the size, yields flexibility that can improve the overall trade-off, as we can see in the average control curves in blue. So on the more substantial data sets, ImageNet and PlantClaf, we see that there's a significant gap between the blue method that do average control and the methods that just do it on a point-by-point basis. Now, I mentioned calibration before, and that's because I wanted to make the point that these methods that trade error rate and set size really rely on calibrated output probabilities, because you're using probabilities from the model to set appropriate thresholds for deciding which inputs, or which classes, that to include in your sets. And so the set valued setting is actually a really interesting testbed for model calibration. And as we can see in these plots from that survey paper, if the method were to achieve this kind of safety guarantee where we have a theoretical constraint on the point-wise error rate, and then we actually measure the error rate that we achieve, we would be tracking this red line. We see when we use the original probabilities from a classifier that hasn't been calibrated, we see that the median is well below the point-wise error rate, but the error bars, particularly the 10 to 90th percentile, exceed the error bars, exceed the theoretical constraint. However, if we just do a simple calibration of probabilities, we could get our error bars mostly within the theoretical constraint. And if we do an additional correction when we do the recalibration, we can have the bars pretty firmly within the theoretical constraint. So we can actually offer these sort of safety guarantees on the risk with these models. Again, this is the probability that the true class will not fall within our predicted sets. Okay, so let's talk about PlantNet 300K. This is a really nice data set that captures a lot of the characteristics of biodiversity problems. It studies plants at the species level, and there's strong class imbalance, as you see in this plot here. So you see that effectively the 80% of the species, the ones with the lowest number of images, account for only 11% of the total number of images in the data set. So it's very, very underrepresented, where a few species really dominate the training data set. And I'd also like to point out that there are visually dissimilar images belonging to the same class. This problem tends to be tackled in mainstream computer vision, different views of objects or different types of objects. But here, this problem on the right are examples of visually similar images belonging to two different classes. So two different species that look basically the same to the human eye. And this is this example of the aleatoric uncertainty that even if you're a trained botanist, you have difficulty differentiating between these two different species. And so this is a problem that doesn't tend to show up as much in mainstream computer vision. Now, when we measure performance on PlantNet 300K using typical metrics like top one accuracy, you actually see that various confidence and vision transformers are doing reasonably well. And that's because it's using a micro average. It's basically averaging over all of the examples in the test set. When you do a macro average, that means you average the performance per class and basically treat every class the same. Then you'll see a massive drop in performance. And so you can see this up at the top right here as well. For the classes that have greater than 2,000 images in the training data set, the mean accuracy in that bin is upwards of 90%. But when you look at the classes, the species of plants that are underrepresented in the data set, you have a much lower accuracy rate. And so the other thing I want to point out about this data set that was explored when it was released is that in a lot of mainstream computer vision data sets like ImageNet, models are, and have been for some time, surpassing human level performance. But that's not the case here on PlantNet. If we compare the performance of CNNs here, we're talking about error. The error for models versus a trained botanist are actually considerably higher. So there's a lot to do in terms of improving performance, particularly across the underrepresented classes. So with one of my students, Kevin Casa, we looked at scaling up some of these set value classification methods to the PlantNet data set. And in our observations, we had a similar one to the survey paper in that average control, allowing for this flexibility in set size across the data set, generally allows for improved performance. And I'm going to just move my video up here because it's a little bit hard to see these results down here. So while the VIT models might come better calibrated off the shelf, a carefully optimized training recipe that we find in the form of the data efficient image transformer called DEET, it actually yields pretty huge gains in terms of macro average top 1 and top 5. And so these macro averages are the ones that I mentioned before, where it's actually doing the averaging of performance within the classes and then doing a final average. The top one is the one that kind of masks the fact that you're doing very well in the dominant classes, but doing quite poorly on the underrepresented classes. And then over here, it's a different type of measurement. So this is used, this average 5 accuracy is used for average variable size set value classifiers, which output between, for example, 1 and 5 classes. And so here, we're measuring the accuracy on these variable size sets. And again, doing a macro averaging where we're looking at it at per class level, but we actually see improved performance when we measure it this way. We allow the model to output variable size sets, but we don't measure it on the sort of top K. We actually use the complete set to do the evaluation. Okay, bringing my video back down here again. So with Kevin, we've also been exploring this alternative notion of building set value classifiers that comes from the frequentist statistics community. These are a set of methods called conformal prediction. And these methods turn heuristic notions of uncertainty, like the maximum value out of the softmax output, into reliable ones through a post-training adjustments. And they can be used to build set value classifiers that have these safety guarantees around risk. But the methods have, to date, have had sort of taken some time to gain traction in the machine learning and computer vision community. And so they have remained largely untested in modern architectures like vision transformers, and under the settings that we typically find in biodiversity, like train test distribution shifts and long tail label distributions. So we've been spending some time focusing on how these methods actually perform under these settings. So there's a variety of different methods for conformal prediction. The framework is actually quite general. While it can be used for to build set value classifiers, it can also be used to produce regression models and be used for time series predictions and all kinds of other machine learning tasks. We've been focusing on classification, and we've been focusing on these 4 methods. So the threshold conformal prediction method is very much like the baseline method we considered before. It constructs confidence sets by thresholding probability. So anything beyond some probability at the softmax, those classes get thrown into the set, and that threshold is chosen based on a held out calibration set. Adaptive prediction sets, or APS, they construct confidence sets using cumulative ordered probabilities. So as you start with the highest, say, softmax output class, you add it to the set, and you keep on adding classes according to that sorting until you've exceeded a certain amount of probability. And again, that amount of probability, that threshold, is chosen using a calibration data set. And this leads to more robust coverage at the expense of larger sets. And then there's regularized adaptive predictive sets, which is a small change to the APS method, but it tends to reduce the set size to make it more comparable to the ones produced by THR. And then, while the methods 1 to 3 are post hoc or plug-in, they only happen after a model's been trained, you can actually make the model training procedure aware of that it's going to be going through this post hoc calibration process, and you optimize the conformal prediction process during learning. So this is called conformal training. We've tested several different architectures in our experiments, of course, ResNets, as well as various types of vision transformers. And for those of you who are familiar with VIT, you'll know that in the original paper, to match the performance of CNNs, you had to use a larger data set, namely ImageNet 21K. But subsequently, the data efficient image transformer paper has shown these are also VITs, but they trained only on ImageNet 1K using a very carefully designed training procedure, various enhancements to the training paradigm used in the standard VIT model, and have shown that you can actually get CNN-like performance, or even exceed that a bit, without needing to change the training data set. So we look at a couple things. So one of them, as I mentioned, was the performance of these conformal prediction methods under distribution shift. And we adopt ImageNet 1K as the training data set and explore several test set alternatives. These test set alternatives include ImageNet V2, which is basically a new ImageNet test set that's expected to follow a similar format and collection process as the original ImageNet, so minimal distribution shift. ImageNet C for corruptions. These apply common visual corruptions to the ImageNet validation set. In this study, the Gaussian noise, motion blur, brightness, and contrast corruptions were investigated, and we evaluated these different types of corruptions. We considered ImageNet A, which are naturally adversarial images that a ResNet 50 incorrectly classifies, but can be correctly classified by humans, and also ImageNet R, which consists of rendered versions of ImageNet classes like drawings and cartoons, etc. So our key result here is that the performance of the conformal prediction methods degrades across various neural architectures when tested on distribution shifted ImageNet datasets. So we're targeting a coverage that is the probability that the true label is included on average in our predicted set. This holds when we stick to the standard ImageNet training set. But as soon as we make even a tiny distribution shift, we're no longer able to maintain this coverage guarantee. So this is basically, again, we've set the coverage and calibrated based on regular ImageNet test set, but when we flip to a different test set and measure it, we lose our coverage guarantees. This is even more severe when we try adversarial or rendered images. And another observation that we make is that the modern architectures, like the data efficient transformer and vision transformer, they actually do much better in terms of their average set size, called the inefficiency. In general, the ResNet models are doing quite poor. So this seems to be consistent with what Guo and Minter and others have noticed about poor calibration in CNNs. The other phenomena that's present in biodiversity that we looked at is in the long-tailed label distribution setting. So a few things to note when we assess on the PlantNet 300K long-tail data set. We again aim at 0.90 marginal coverage, and we see whether we use CNNs or data efficient transformers or vision transformers, the coverage guarantee that we aim on averaged over the entire data set is actually maintained. But when we dip into looking at it on a per class or class conditional measure, we notice a significant drop between the guarantee across the data set and the guarantee on a class per class basis. The other thing I want to point out too is that similar to our other experiments, the data efficient image transformer that uses a careful training recipe is also performing considerably better than the CompNet or the standard vanilla vision transformer. So both on coverage, we're noticing sort of this drop when we look at it per class level, and that's something that we had not investigated before. We see this with the average set size, also called the inefficiency. Again, at the overall level, when we average across the entire data set, where that's dominated by the frequent classes, we do decently well across the different classifiers. But when we look at it at a macro level per class, we see some considerable jumps in terms of the average set size, and it's just horrible for example, the APS classifiers. I'd like to tell you a little bit about how the DNA barcodes and the imagery get linked up, some possible projects on the horizon as well. So I'm doing a collaboration, as I mentioned, with the Life Plan project led out of Finland and Sweden and Duke, and we have been working with these bulk images where the insects have been collected from malaise traps. And the idea here is to detect and count the different arthropods from the images. And the interesting aspect of this data is that we have metabarcoding. So this means the entire sample, basically a jar of insects, have been run through the DNA barcoding pipeline. And we have barcodes associated with everything that showed up in that particular sample. So this leads to an interesting vision setup where we actually have a varying number of possible classes for each image, and these are verified using the barcode. And we've been using the Toronto Annotation Suite to do segmentations of the insects using their AI-assisted segmentation tool, and also working with expert entomologists to do a hierarchical labeling. There's also the potential of utilizing both the bulk imagery and the high-resolution K-ONS imagery of individual insects, because in some of these bulk insect images, the annotators actually clipped out the actual insect or taken out the actual physical insect and run it through an individual insect imaging pipeline. At NeurIPS 2021, I met a group at Purdue who are actually utilizing the DNA barcodes. It's the first time I'd seen this in sort of the mainstream ML literature, combining barcodes and imagery to do zero-shot classification, where the DNA barcodes were actually the side information for the image-based classifiers. And this model can classify samples of both described and undescribed species. The described samples are assigned a species label that has been observed in the training data, whereas the undescribed samples are assigned a genus level, which is the next step up in the taxonomic hierarchy. And this is an advancement over standard open set methods, which basically take something that is unseen in training as to be just flagged as an outlier. So the stack is actually quite simple. At the top, you'll see that they have a ResNet-based embedding pipeline from the images. And also every image has an associated barcode. This is run through a 1D CNN. So we get embeddings for both images and the DNA barcodes. And so they learn to map the ResNet image features to DNA barcodes by using this transductive ridge regression approach. And the fact that it's transductive means that you're using unlabeled test data to be able to map image data with labels into DNA feature space and combine this information with DNA embeddings. And so they have a hierarchical Bayesian classifier that at test time can accept unlabeled images, and a test sample can be assigned to one of the described species, as I mentioned, that we're seeing at training time, or assigned to an undescribed species that is flagged with its genus, again, the next level up in the hierarchy. And so what they're able to show, I think I'd say take note that even on scene classes, an image-only classifier being able to classify to species is very poor compared with a traditional bioinformatics pipeline. So this is just using the barcodes alone, which can just recognize scene classes at a very high rate. Not as good on the unseen classes, but still considerably better than image only. If they do zero-shot Bayesian classification just in the DNA space, they get a certain level of performance. But the main point of this paper is showing that they can use image-based input to improve over DNA alone, where if we're looking at sort of the harmonic mean over the seen and unseen performance, it's better than using either modality alone. So this is actually quite promising and very exciting for us as we have access to both imagery and DNA data. And I just point out that their database is actually relatively small, 32,000 insect images from about 1,000 described species. And we've been actually working on a project lately where we're building a data set that has approximately 1.1 insect specimens from malaise traps that are imaged with these K-amps microscopes. So these are the different orders that are present in the data set, along with the number of samples associated with those orders. Of course, you can see it is highly unbalanced, as we mentioned before. But this data set includes the taxonomic classification to the level to which an expert annotator is confident. It also has the full DNA sequence barcodes, and then it also has this barcode index number, which again is a species proxy that results from a clustering algorithm run in barcode space. And so we provide order and family level classification baselines, but we actually think it's going to be most interesting for taxonomic classification methods that deal with sparsely annotated trees. And so I'm going to wrap it up here. ML for biodiversity yields many interesting technical problems that inspire us as researchers. And ultimately, apart from the interesting machine learning problems, our goal is to derive desired future behaviors in the ecosystem. Everybody cares about biodiversity. Everyone's mindful of biodiversity. We're appreciative of the natural capital available. We consider the future. We measure biodiversity regularly. And we collaborate to inspire and advance biodiversity action. Thank you.